Hi, I'm Caitlynn. My internship was at the Aquatic Germplasm and Genetic Resource Center. My mentors were Dr. Tiersch and Dr. Gutierrez-Wing, and I was tasked with developing and conducting my own research project. And this is what I came up with. It's the effect of NAA, zeatin, and natural microbial assemblages on Isochrysis galbana growth. And that's my outline. A little bit of background. There are many uses for algae, and one of them is what I like to call the 3 Fs, which is food, fuel, and fertilizer. And it's also used for pollution control and like wastewater treatment plants. At the facility, they also do a lot with cryopreservation, whether that be with plant or animal tissues, including algae. And that's just to preserve the genetics for future use. The species I'm working with is called Isochrysis galbana. It's a marine species of microalgae that has flagella, and that makes it easy for me to see under the microscope. And that's what it looks like. And the 2 additives I'll be using is naphthalenoacetic acid and trans-zeatin riboside. Both of these are plant hormones that promote growth through cell division. See, and this was my experimental design. The algae alone, also known as an exinic culture, and the algae plus bacteria, also known as a non-exinic culture. And I either have no additives, additive 1, the NAA, or additive 2, the zeatin. My methods to isolate the algae, I actually did serial dilution, as you can see here. And from that serial dilution, I put it into a 24-well plate, like in 1 milliliter in each. And I put it in the algae cabinet, and it stayed around 29 degrees the entire time, and it received light 24/7. And I also had a little bit left over from the dilution, so I also put it into some flasks. Okay, and what I did, I would pipe it out a little bit each time and every day, and use the microscope, which is 400 times, and I would count how many algae cells. I would have to vortex it first, because algae tends to stick to the bottom. And as you can see here, on the left side of the screen, that's the exinic culture. You can see there's not very much bacteria in the background. And on the right side, you can kind of see in the background there's a little bit of bacteria. The yellow circles are the algae cells, and the red arrows are pointing to the bacteria in the background. And the arrows on the left side are pointing to the little flagella that you can kind of see. So these are my results for the well plates. The exinic culture did not grow as well as the non-exinic culture, and that was kind of expected. So it shows that bacteria and algae kind of have a symbiotic relationship. And the same goes for the flasks. And I found it strange that the additives did not help the growth in the exinic culture, but it did in the non-exinic culture. So I'm thinking maybe that the bacteria also helps with taking in the nutrients and the additives. So this is my conclusion. Yeah, so yeah. Bacteria plus the additives are good for algae growth. Yeah, also, I forgot to point this out. If you notice in the well plates, the growth for the exinic culture was a little bit higher than it was in the exinic culture for the flasks. This is because there was some contamination over time, so here's some free advice. Don't put everything in the same well plate, especially if you're trying to keep it sterile. Some future research opportunities. Try culturing it in the best conditions, maybe culture from the best well plate. I'm hoping to do that this week, actually. See if this works with other species of algae. Try cryopreserving this algae and see if the bacteria has any effect on the freezing process or the additives. And maybe try to determine what strains are in the algae and maybe which ones are bad and which ones are good or which ones have no effect at all. Some side projects I did while I was there. I built a fish tank from scratch. Well, not a fish tank filter. It's an undergraduate filter. And that's it, in use. I also got to play with liquid nitrogen a little bit and learning how to cryopreserve algae. So that was actually really fun. I also learned how to 3D design and print. So I printed myself a dragon. Any questions?